I'm going to talk about immunofluorescence and immunoelectronmicrography. First, I'll take up immunofluorescence. Immunofluorescence is a technique which is performed using fluorescent molecules which are conjugated to the antibody, and the antibody is used for finding the corresponding antigens. The fluorescent molecules generally used are fluorescein isothiocyanate, which absorbs blue light at 490 nanometers and emits intense yellowish green fluorescence at 517 nanometers. The second molecule that is used is rhodamin, which absorbs light at yellow-green range of 515 nanometers and emits deep red fluorescence at 546 nanometers. By using two different antibodies which are conjugated to two different fluorescent dyes, one can do differential staining of the target tissue where the two different antigens to be identified. I will explain about this differential identification of two different antigens little later. We will first consider the simple technique of using a primary antibody for the recognition of an antigen by direct immunofluorescence test. Here let us take the example of a bacteria which has antigens on its surface, and a corresponding primary antibody is available for these antigens to be identified, and one need to prepare a slide of the bacterial preparation, and the bacteria is immobilized on the slide. The primary antibody is conjugated to a fluorescent dye, either fluorescein isothiocyanate or rhodamin, and this primary antibody is added to the slide where the bacteria is immobilized. After adding this antibody over the surface of the bacterial preparation, allow it to remain for some time, then wash off the excess of antibody, and then visualize this slide under the fluorescent microscope. When you visualize, if you see the fluorescence, it indicates that the antibody which is labeled by the fluorescent dye is bound to the antigen that you are trying to detect in the particular bacteria. Similarly, same direct immunofluorescence technique can be used for various cell preparations. For example, if one need to identify a specific antigen in a cancer cell preparation, provided the antigen that is expressed by the cancer cell has corresponding antibody with you, you label the corresponding antibody with fluorescent dye and add to the preparation of the cell smear which is smeared on the slide. When the antibody is added to the cell smear, it will bind to the antigens if the antigens are expressed by a particular cancer cell. And after sufficient washing, when you view this slide under the microscope, if you see the fluorescence, the antibody is bound to the antigen, and that gives the fluorescence because the antigen is expressed by the cancer cell. The second technique is slightly different, that is indirect immunofluorescence, where the preparation of the cell or a bacteria, target bacteria, is prepared on a slide and the primary antibody corresponding to the antigen is added. Here, the primary antibody is not labeled with any fluorescent probe. Primary antibody could be obtained from the source, that is the person who might have produced the antibody against the infection of a bacteria, or who has the antibody against the certain cell antigens. And after adding the primary antibody, the preparation needs to be washed, given a wash. Since the antigen antibody interaction is very strong, the primary antibody does not get washed off. After the sufficient wash and excess of primary antibody is washed off, the secondary antibody, which is labeled with the fluorescent dye, is added. The secondary antibody is nothing but it is an antibody against the primary antibody, which you can call it as anti-globulin or anti-antibody. This secondary antibody is conjugated to fluorescent dye. On addition of the secondary antibody, if the primary antibody is already bound to the cell antigens and it is available there, the secondary antibody will recognize the primary antibody and bind to it. If the primary antibodies are not bound to the antigens, then the secondary antibody will not be able to bind to anything. If the primary antibody is bound to the cell antigens, the secondary antibody will bind to the primary antibody and give very good fluorescence. The indirect immunofluorescence technique is much superior compared to the direct fluorescence technique, because the primary antibody molecules will bind to several secondary antibody molecules, because of which the fluorescence will be much brighter than that you observe in the direct immunofluorescence. The differential staining technique that I had explained earlier or told earlier is nothing but when you use two immunofluorescent dyes simultaneously at the same time. For example, if there is a lymphocytic population and you have prepared a smear of lymphocytic cells, in order to detect whether this lymphocytic population is expressing two different antigenic receptors, say for example CD4 receptor as well as IL-17 receptor on their surface, one need to have the secondary antibody which is tagged with two different immunofluorescent dyes. Say for example, one secondary antibody meant for IL-17 as shown in the figure is conjugated to fluorescein isothiocyanate, and the other secondary antibody is conjugated to rhodamin. The primary antibodies against these two receptors need to be added first, and after giving a sufficient wash, the secondary antibodies with respective fluorescent dyes need to be added. After some time, after a sufficient wash, when you observe the slide under the fluorescent microscope, you would see two different types of fluorescence, because two different antibodies to recognize two different antigens were added, and these two antibodies were conjugated to two different fluorescent substances, which give fluorescence at two different wavelengths, giving rise to two different colored fluorescence. This can be visualized in the slide here. CD4 is recognized and it is giving rhodamine fluorescence, and IL-17 is recognized here and it is giving fluorescein isothiocyanate fluorescence. So in this way, fluorescent probes conjugated to antibodies can be used for identifying antigens at cellular level in the tissue preparations. Specific antigens expressed by some cancers can be also detected by this immunofluorescent technique. Bacterial antigens of different species of the bacteria can be also identified by differential technique. I will move on to now immunoelectronmicrography. This is another technique where the antibody molecule is going to be used in the electron micrography by conjugating it with another protein which is called as ferritin. Ferritin is a protein produced by bacteria, and this protein can be extracted from the bacterial preparation. Bacteria produce this protein to attract iron, and they need iron as a micronutrient for their growth. Advantage of this ferritin protein can be taken by conjugating this ferritin molecule with monoclonal antibody produced against any cellular receptor or any cellular antigen which is specifically expressed by certain cells. Say for example, you are trying to carry out electron micrography of a particular cell preparation, or say for example cancer cell preparation, and you are trying to identify the expression of a particular antigen by that cancer cell. If that antigen is expressed by the cancer cell, probably that cancer cell will become radiation resistant. To find out whether that cancer cell has expressed that antigen, you would like to carry out this technique. So the monoclonal antibody prepared against that antigen is conjugated to ferritin protein. The cell preparation is prepared on a slide, is prepared in the sense you perfuse the cell or the tissue, cancerous tissue, with monoclonal antibody conjugated with ferritin. After some time, you perfuse the tissue with iron solution, which could be ferrous chloride. The ferritin molecule attracts iron and binds to it. After some time, perfuse the tissue sample with normal saline to wash off excess of antibody and other ion molecules. Now, you prepare the tissue sample with a conventional procedure for the electron micrography. And it will be seen that because of attraction of ion by the ferritin molecule, those areas will become electron dense and appear as black spots. If the antigen that you are trying to detect in the cancer cell is expressed, the monoclonal antibodies will have detected that antigen and will have bound to it, and since they were conjugated to ferritin protein, they would attract ion and make the area electron dense. Hence, in the electron micrography, you can see on the left hand side on the cell surface, the black spots have appeared, indicating the appearance of the antigen in a particular cancer cell. Similarly, the cell cytoplasmic proteins can be also detected using monoclonal antibody approach, using ferritin as a molecule that conjugated to monoclonal antibody, and electron micrography can be carried out. This is how the immunoelectronmicrography can be performed, and the fine antigen expression on the cell surface or within the cellular components can be identified.